we now go into our next session on microbiome, animal health, and bioagriculture. This session will be chaired by Mats Bjørnvad, Senior Manager for Microbiomics and Microbiome Discovery at Novozymes, Denmark. Mats was trained as a molecular biologist and has worked with genetic engineering for the most part of his career. Since 2017, he has headed Novozymes Denmark's R&D activities around microbiomics and discovery work in relation to probiotics, especially for humans, animals, and plants. And as Mats joins me, I'd like to remind you that you can ask questions to our speakers in the Q&A tab at any time, and we will be saving all questions to the discussion and Q&A part of the session. So warm welcome, Mats, and the floor is yours.
Thank you, Sarah. Thank you so much, and I'm very happy to be here and host this session and chair it for our distinguished speakers, Professor Tom Gilbert and Professor Lars Hesbjerg Hansen, who will come and help us out and try to understand better the complexity, the association between large animals, humans, and plants, and how to analyze and study that. At Novozymes, we have a purpose, and that is to find biological answers for better lives in a growing world. And that talks directly to the understanding of the complexity of the biology and how it interacts between microbes and large animals. And as humans as well, it's very important that this is in balance. And then this purpose is supported by our strategy, which is called Better Business with Biology. So that is simply to secure that we make products that are based on biology and that can have meet a need in the market and have an impact in the market. And for years we've been working with enzymatic solutions. Doesn't really click. Can you move to the next slide? Okay, so for years we've been working with microbes as solutions for our customers and the consumer, and we also work a lot with enzymes to be brought to our consumers. And if we look at the United Nations Sustainability Goals, a lot of what we deliver actually supports the development of these goals. For example, for supporting sustainable food production and resilient agriculture practices, this is where our microbes and enzymes can play a role to secure that we get high yields from the crop using less chemicals and less energy in the production. Also, using fewer chemicals in this field secures that we have more clean water at the end. But also cleaning water for waste water plants and other types of water cleaning, we deliver enzymatic and microbial solutions in this field. For enabling a low-carbon future, we of course have been the leader in the field of developing enzymatic solutions and microbe solutions to take sugars and turn it into ethanol that can be used as biofuel to substitute fossil fuels that is otherwise generating a bad impact on the carbon and the climate changes. So if you look at some of the concerning numbers we have on the planet, the 10 billion people we will be around 2050. Will be 10 billion people on the planet, and that creates a huge pressure on the planet and the needs for food and fuel and so forth. And actually the 70% of extra food that we need to produce in 2050, how do we do that in a sustainable way? We have to use biological solutions. And this is again where we need to understand the complexity of the biology and translate that into solutions that can help us in the future. And of course the 1.5 degrees C that we need to secure that we don't have a high temperature on the planet, keep that around 1.5 degrees Celsius is of course something that we help by delivering products that can turn down the use of energy and CO2 generating processes. We deliver that in many fields. One of them is also by lowering the temperature for laundry washing in households. But moving on, we're not only looking at industrial processes and supporting that, we also have a new growth pillar in the company. It's called the Novozymes One Health. This is where we're addressing the needs of human health. And we have just recently bought a company in US, Microbiome Labs, and a company in York, Ireland, Precision Biotics. And both of these companies have excellent product portfolio They know how to run clinical trials. They know how to market these probiotic solutions for human use, in gastrointestinal use, for immunohealth, in skin health, oral health, and even in the cognitive health. We have products that supports the gut-brain axis, for example. So these are some of the excellent examples we have for human health. Then recently, now we're changing gears and moving more into the microbiomic space, which is the topic of today's event. And here recently we acquired a technology platform from a small startup in San Diego called Biota. We acquired the tech team, the data science team, and a technology platform simply to boost our competencies with our already existing microbiomics researchers, but also boosting the data analysis capabilities and the data management. So this will increase our capabilities to and support the microbiomics-based research that we're doing for human health, animal health, bioagriculture, and plant research. So this is going to be a huge change and improvement of our capacity to do multi-omic studies, which is what is needed for the future to find new biological solutions that are based on microbiomics research. So talking about a little bit about how we see microbiomics-based research and how we use it in our part of the industry. So for us, microbiomics-based research is to fuel and feed a growing world, because by using microbiomics research, you can actually go into discover of new biological solutions. I'll come back to how we actually approach that. Or you can actually study the effects of our biological solutions when they have been put into action in an animal, human, or in the soil of agriculture. So those two different approaches we have, and both are equally important. But let's zoom in on how we actually do for discovering a new biological solutions, because this is highly interesting. So we have two ways of doing it. The first is a more traditional screening-based approach, where we actually screen our huge culture collection to identify the microbes that can make a change and have an impact in the application. So we screen it for various parameters that is relevant for the application, and to have the efficacy that is needed in the application. That's a screening-based approach. That's a more traditional approach. But if you then go to the right, the microbiome-based approach, this is where we look at the discovery approach, looking at a population of a plant population or cow population in this example, and then study the microbiomics, the multi-omics, what are the microbes producing, and how does the animal react to this in terms for cows, for milk yields, methane emission, and so forth. Then try and find those microbes that are actually driving the high-performing host, and that is very different from what it looks like in the low-performance host. So that's the delta microbiome we're looking into to try to predict some of the single or consortia of microbes that actually are driving the performance of a subpopulation of a larger population. And then that goes back to, is being fed back to that understanding is being fed back to our culture collection and screening, and then we pull out these strains that looks interesting as a performance enhancer or health enhancer in the animal or plant or human. And then we screen for that, pull that out, and put it back to test it out in animal trials or plant trials or even human studies. So that's an iterative approach, and that's a very powerful way to actually use microbiomic-based research understanding to drive new developments of new solutions. And it's not only the microbes, it's also their genes that could encode new exciting enzymes that prove potent against pathogens, or to degrade certain fibers that could be nutritional and generate a healthier gut environment. So that's a little bit of a view into how we from the industry look at microbiomics-based research and use it in our endeavor to deliver biological solutions to make sure we have a sustainable living on this planet that we need to take care of, as being 10 billion people on it pretty soon. So turning over to the presentations, Tom Gilbert will give a presentation and teach us more about the association studies of animals, humans, and their microbiomes. And I'm sure he'll also come with arguments for the personalized microbiome treatments. And I'm looking very much forward to learn more about that. And Tom Gilbert, I met last year in relation to a grant around the hologenomics. And when I read that grant, I was very surprised with the visionary and huge impact it could have. But it was very visionary, and it just shows Tom's great passion for driving science around very complex matters. So that will be a very great walkthrough and presentation from Tom, I'm sure. So please join us, Tom, for your presentation. Let's just push the green one there.
Thank you. Great. Well, it's a really great pleasure to be here. Thank you so much for inviting me. So I am going to talk about an idea, really. It's about a belief that we have the technology today to think about how we use microbiomes in a different way. And so my title, Applied Hologenomics, Should Microbiome Treatments Be Personalized? So just for the record, I am not a microbiologist. I'm terrible at that. I'm an organismal biologist, and most of my career has been working on the big end of things, and I largely ignored microbes for a long time. So a very good question you should be asking is, why the hell am I talking today? You know, what can I possibly offer? And so I'll try and hopefully give some reasons for that in a minute. I am the director of a research center called the Center for Evolutionary Hologenomics, and our focus is on what's called the Holobiont or the hologenome. And I'm really on a bit of a mission, a crusade right now, to actually ask people to consider including the Holobiont and the hologenome into their thinking. So in light of this, I've got three aims to cover. Firstly, I'd like to clarify what a holobiont is and what its hologenome is. So it's a terminology thing. Secondly, I would like to present an argument that maybe we should view the microbiome as a phenotype conditioned by host genetic variation. Just like size or color or fat production is a phenotype, we actually don't view the microbiome as anything more than a phenotype that is ultimately constrained by host genetics. And then if this is the case, what I'd like to briefly discuss is the possible implications of that for applied microbiome research. So firstly, this term holobiont, well, a very easy way to explain it is through this picture. This here is a vulture who I'm sure many of you are aware of. Vultures have evolved to fill the niche of eating really nasty, rotting carrion that by all rights should kill the animal straight out. And a classic evolutionary question is, how have they evolved not to die when eating these horrendous, horrendous things? And a classic organismal biologist like myself would say the answer's in the genome. Let's sequence the genome of the vulture and let's look in the genome for genes that somehow allow it to do this. And in fact, people have done this. But actually, my colleague Lars, who is talking up next, actually ran a study a few years ago that actually looked at the microbes in the vulture. And their study showed, you know, these microbes actually seem to be doing something functional to do with the nasty bacteria they're encountering. And that work was taken further by Lysandra Zepeda here, who was a PhD student at the time. She sequenced the microbiome of the vultures, and she found that there was all sorts of particular microbes present with particular genes that did things that helped it actually deal with the very nasty things it was eating. So really what we're looking at is not just how did the vulture get to fill this niche, but it's a holobiotic basis of adaptation. The vulture is doing something, the microbes are doing something, and together they are filling this role of things. So it's a union together. And this comes down to the terms. The term holobiont basically says you can't understand the animal without its microbes, and you often can't understand the microbes without the animal. So let's stop looking at them independently and look at them as one, and this is the term holobiont, the union of the two. And the hologenome is simply the total genes that are present in the system. So the genes of the vulture, the genes in the microbes that together help it fulfill this niche. Now, actually, to most people here, it's probably obvious. If you think this way, holobionts are everywhere. There is almost no life form that we know of that actually exists without microbes, right? You know, they have them on their roots, on their leaves, in their guts, on their skin. Clearly, life is built around the holobiont, but we couldn't study life in this way for a long time because of technology and limitations in sequencing potential and computational potential. But now all of that is changing, and that is why we are advocating as much as we can put the two bits together. It is possible now, the tools are available. So this center that I direct, the Center for Evolutionary Hologenomics, we have a number of questions we're interested in. And one question is just, what are the myriad of ways that a microbiome might shape its host life? Now, of course, there are the very, very obvious ways. Microbes and their relationship with human health is a very, very big topic right now. Microbes and animal production affecting how they grow or the quality of their meat or how crops grow. Microbes are everywhere in the applied context, and we know that's because microbes basically process foodstuffs into the things bodies need to grow. No big surprises there, but what's more interesting, when you start looking into the sort of ecological literature, the basic biology, we find more and more examples of really remarkable traits which can be explained by the microbial action on the host. So not just nutrition, but things like toxicity resistance, whether a certain plant kills this rodent in the top right corner or not, depends on its microbiome. There are studies showing, for example, hibernation in bears is driven partly by the microbiome, and without the microbes, they wouldn't enter hibernation. There are studies showing thermoregulation is affected, studies showing behavior is affected. And once you see this incredible range of things that microbes do, you can start to realize if we really want to understand how life works, we have to factor in both parts. We just cannot look at one alone. Now, that is one basic question. In many ways, it's stamp collecting. It's just, yes, we found another explanation and so on. But maybe more intellectually satisfying is the question, what actually shapes the microbiome in the first place? What determines which microbes are inside the host? And there is an extremely popular answer to this, which is the environment. There are plenty of very, very big papers and plenty of studies showing the environment has an enormous, enormous effect on the microbe. And it's very, very true. If I decided to become vegetarian and move to Thailand tomorrow, my microbiome would change instantaneously, right? But that doesn't mean that the host genome is irrelevant. And going back to my original aim, we are very strong believers that we have to factor in the role of the host genome if we ever want to realistically understand the relationship of what's going on. It may not have the broad changes at any particular instant, but it's got to be somehow constraining the system. So this raises the question that lies at the heart of what we are trying to work on right now. And does the genome shape the microbiome, and exactly how, and what kind of limits does that bring in? Now, I should say, on one level, the answer is obviously yes. If you consider this figure, it shows 3 different kinds of animals based upon their gut. We have the monogastrics, we have the ruminants, we have the hindgut fermenters. They have very, very different gut structures. And of course, their gut structure is determined by their genome. So there is no doubt that the genome of these different species and how these genomes vary affect the inside of the animal. And by affecting the inside, they affect the microbiome. So not particularly surprising. It's much more controversial when you move to the intraspecific level, the variation within species. And this is partly due to the fact that many of the studies into this have been performed on humans. So this is just one example of a nice study from about 7-8 years ago now. Different groups of humans had their microbiome sequenced, and what the gray plots here are basically is a standard network of relationship of microbes, and the colors relate to which microbes are present where. And if you look in the bottom right, where you can see the Hadza hunter-gatherers from Tanzania, their microbiome is quite different to, for example, the top right, which is Scandinavians. But of course, the problem is they're living in very, very different environments, eating different food, right? So we can't really use this to say anything about genetics. It's just look at the effect the environment has. There have been studies, very elegant studies on twin cohorts. The rationale is when you raise twins together, the environment is somewhat constrained. And when you compare the microbiome and how it varies between identical twins and non-identical twins, where you argue for each pair the environment is stabilized, is similar, you do actually start to see overall a closer similarity in the microbiome in the identical twins than the non-identical twins. But again, the problem with humans, we have too much free will. For all we know, Ashley and Kate Olsen there on the left, maybe one's a vegetarian, maybe one's a carnivore, we don't know, right? So humans, that free will is not a great thing. So where we come in, we organismal biologists, we're coming in from the natural world, and we've been trying to look at systems where we were studying them anyway, studying population structure, but we could tag on a microbiome study. And just one example, this is the work of Kathy Feets, who was a PhD student with us a number of years ago. She was working on sandlance. You can see one here. There are these small, very economically important fish caught across the Baltic. And she was studying the population structure using classic population genetic methods in her thesis. And in fact, you'll see on the map here, there's a green line. That basically separates where the water salinity off the Danish coast changes. It goes from high salinity on the west side and low salinity on the east side. The pie charts just show a representation of the genetic structure of the sandlance. And so we could see there was a clear break where the salinity changes. But what Kathy also did, she was sequencing the microbes in the guts of these at the same time using, back in those days, 16S. And what we show here at the top is a pie chart representation of the different microbial groups in them in A, that is the high salt region, and B in the low salt region. And you can see where that green barrier is, there's a big difference in microbes. But of course you can say, well, yeah, Tom, the environment changed. We had high salt and low salt. Fortunately, she had a water control and that wasn't changing. So what we're essentially seeing is, although the microbes are constant, that change in the genetic structure seems to be mirrored somehow in the microbial structure. Now, there are many reasons for this, but things like this start pointing towards situations where maybe genetics is having quite a big effect on what is going on. And bear in mind, this is all the same species of fish, very, very closely related, just slight population structure in there. So our view is very much that we should be viewing the microbiome as a phenotype, and we believe to some degree that phenotype is being constrained by the host genetics. Now, I want to try and put this into pictures, and this is the slightly abstract bit. Let us assume we just made a catalog of all the microbes that might exist, and assume this picture is the catalog. It's just all different microbial lineages laid down next to each other. Proximity is irrelevant. It's just box 1 is one microbe, box 2 is another. The color here is whether in any situation a microbe is present or absent. So we have some very dark blocks. That's where there's a lot of a certain group of microbe. We have some white ones. The microbe is not there for whatever reason. If we just imagine we know all the microbes, you could define what microbes possibly could live inside a host. So we have host A, it could be me. That red box might be defining all the possible microbes that could live inside me. They may not live inside me. I may not right now be smoking marijuana or eating fermented products or whatever. So they may not necessarily be there, but they could be there. The ones outside the box could never be inside me. Some kind of deep water hydrothermal vent microbe would just not survive inside me. So my genome is controlling which could possibly be inside me. Now, of course, as I said, the environment can change everything radically. So if you look at the box now and we change the environment, the microbes inside me might be completely different, but they are still constrained within that space. What we're interested in is if we compare the genome and the potential microbes of two individuals, what is the size of the overlap? Is there a lot of overlap? Is there no overlap? Here we have one individual in green and one in red. All the unique bits to green only are areas where only B could benefit from a microbe because they just can't live inside A. And this essentially is what we are trying to measure. Is there overlap? How much does the genome control it? And are those microbes, which are actually exclusive to one or other genotype, actually important in any way? Now, to me, this shouldn't really be very surprising. If you consider many other phenotypes, there's clearly an interplay of genetics and environment. So here's a picture of me and my 3 clonal brothers. We are very, very, very similar. People mix us up. But we have a very big difference in height. So genetically, although we're very, very similar, because of the way we were raised, the food we ate, I was the first child I was never forced to eat. I stayed small. My brother on the other side is massive because he ate constantly and my mum wouldn't let him not eat. So the environment did have an effect, but none of us are ever going to be an NBA basketball player. We will never get to that size because our genetics is limiting it. And none of us will ever be a competitive jockey because we just can't get that small, right? So the genetics is shaping the baseline. The environment is actually giving the resulting phenotype. And this is what we think is going on with microbiomes. Yes, the environment has an enormous effect on the microbiome, but only within a framework that has to be constrained by the genome originally. So if you accept this, or accept this could potentially be true, there are a few consequences. Firstly, there may be some new explanations for some of the old evolutionary biological questions that classic biologists like myself obsess about. What defines success or failure in evolution? What defines which species we can domesticate? What defines which species are very, very good at invading, or so on? And much of our work is based on that. But I also believe the consequence is that we may need to start thinking about tailoring microbiome therapy. If the genome truly does condition the microbiome, it's basically, in my view, no different to what we're thinking about personalized medicine. So what do I mean here? Here's a very, very childish representation of personalized medicine. On the very, very left of the figure, you basically have something that's generating the drug you want to give people. It could be microbes in a factory. You then make the drug, and then you actually condition the dose of the drug on a human. This is well accepted now. It's basically the promise of personalized medicine and sequencing everyone's genome. It's very, very hard to get to work, but the idea is we're not all going to react the same way to the drug, so we have to tailor it to how the body responds. Now, basically, I would argue the microbiome therapy is exactly the same thing, but moving the factor into the body. You're just taking the microbes, dumping them in the body where they make the drug. So it is just personalized medicine. There should be no surprise that different animals or different people will react in a different way to the microbes you give them. And if you don't take this into account, you run into the same kind of efficiencies that you get with conventional medical treatment. So here's one example, the gut-brain axis, very, very, very hot right now. How the microbes in the gut affect the brain and how they all tie together. If you look at this figure in detail, we don't have time for it now, human genetic variation can affect all those parts of the process. It can affect, for example, how we respond to the metabolites that are produced by the bacteria. It can respond to which bacteria can grow inside us and not get ejected. It can affect actually which microbes work together. So it shouldn't be that surprising. But it is very hard to study in humans. So what we've actually tried to do in the last few years, very much thanks actually to open collaborations from companies like Leroy Seafood Group in Norway, but also actually Christian Hansen here in Denmark, we've started working with them on studies where they are studying the phenotypic response of a commercially important animal, for example, salmon or pigs or chickens, to microbiomes, so to probiotics or to different feeds, and to say, well, look, we can see certain microbes have got an effect on the population, but is it a straightforward effect, or do we need to also take into account the genetics of the host? So in a classic study, the Holofish project, one of our first grants with Leroy on this topic, we have a load of fish being farmed in a standard environment. They're given some different feeds. Now, classically, what we would do is just measure the size of the fish after a certain amount of time, and we would look at the microbes, and we would see which microbes are giving the big fish. But what we're doing is we're overlaying on top of this the genome of the fish, and to see, are all the fish reacting in the same way to the feed, and are they all getting the same microbes, or are we seeing differential results? Because basically, if you see differential results, you start to get towards the idea of maybe we should be conditioning the diet or the probiotic or whatever else you're putting on the genotype of the fish. If there is a certain genetic variant in a fish, which means a certain food comes in and gives a certain microbial community that makes it a big fish, why don't we try and give all fish that genotype to make the system more efficient? So essentially, you could hope this would make the overall process much more effective at growing big fish and reduce the number of results that basically aren't showing any improvement. How do we do it? Well, most of our effort is on this. We've built what we call the Hologenomic Framework. In short, we try and generate enormous amounts of different data from every sample under study. We don't just take a sample and sequence its microbiome. We sequence its microbial transcriptome. We sequence the host genome, the host epigenome, the host transcriptome to get all these complicated bits and then try and build the framework to stick it together. It's very, very hard, but that's at least what we're working on now. And we believe the power, the computational power is there now. The methods are there. What's important, though, is sticking the bits together and really getting an efficient framework. I should clarify, I think this applies to any system where you think microbes are going to have some kind of effect that you care about, whether it's farming pigs or chickens or shrimp, or whether actually you want it to make just happier, better cats or dogs. And lots of feeds today have got probiotics added to them, or faster racehorses. Of course, I think it's relevant to humans, although humans are a particularly difficult subject because we rarely have control over the rest of their behavior, so environment becomes very, very important. I think it's equally relevant to plants. If you believe that microbiome treatments will give you healthier crops, the question is, will all plants with different genotypes react in the same way within a particular species? Will we find anything? I honestly don't know. We've been cracking at this for about 2 years right now, and a lot of it's been building it up. We may not. If we're wrong, it's great for the industry because then you don't have to worry about personalizing. If we're right, it might give methods for tweaking things and making them more and more efficient. There are some major challenges. One of them is, I've said I believe the microbiome is a phenotype, but if you think about it, what is exactly the phenotype of a microbiome? Is it which microbes are there? Is it relative abundance of there? Is it which ones are active? Is it which genes are present? Is it which ones are working together? We simply don't know right now. How do we, what is the relevant variation in the genome? Is it regular mutations, or is it in fact epigenetics, or is it in fact 3D structure of the genome? How do we tie it together? Again, that's an open question, but luckily in our center we have a range of molecular biologists and computational scientists and statisticians who are at least trying to come up with the arguments for that. So that was it, really. I'll end there. So in summary, I think there is no doubt to anybody these days that microbiomes are very intricately linked to their hosts. And if you really want to understand life, you have to look at the union of the two, not the individual. And technologically wise, price wise, we are there now. People who are not taking both into account are largely wasting their time, I think. It's no doubt at all the environment has an enormous effect on the microbiome, but that does not mean that host genomic variation is irrelevant. Just always think back to that example of height and environment and genetics. There has to be some relationship. It may not be an important relationship for the question you're looking at, but at least we have the potential to study it right now and see if it is. And if you accept this, I think that microbiome therapy should not be viewed any different to personalized medicine, especially when the environmental variables are minimal. And this is the true thing about farming. And to be honest, when you have 100,000 chickens in a chicken shed, the environment is standard. When you have 200,000 fish in a salmon pen off the coast of Norway, the environment is standard. And in those situations where the environment's taken away, that is where the genome becomes most important. So with that, thank you to the Microbiome Signature Project and Novozymes for having us, and of course the team working on it. And I look forward to any questions later.
Great. Thank you so much, Tom.
Now.
Yeah, please. That was a great a lot of topics and a great presentation. Thank you so much.
Great.
We'll see you back for the Q&A in just about 20 minutes.
Thanks.
Thank you, Tom. Now to the next professor in line, Professor Lars Hesbjerg Hansen, who will talk about deep learning and also the association, to understand the association of microbes and complex larger animals and plants especially. And every time I meet Lars, I think we're going to talk about microbiomes and microbes, and then we end up talking about phages. I'm sure you also sneaked in some slides about phages and how they affect the balance in the microbiome.
Okay, sure did. Yes.
Very welcome. Thank you.
So I will just see how this works. Turn back. How do I move the red one? Okay, yeah. Thanks a lot for giving me the opportunity to present here today. There's a lot of spectators, and thanks to Tom for a very interesting presentation. I wish today to introduce the ideas and some preliminary results that are unpublished from our Novo Nordisk Foundation project called the Matrix, which is a microbiome-assisted triticum resilience in X dimensions. So what I hope to do today is to show you what we're aiming to do in the Matrix, and and in the collaborative crop resiliency program, which Matrix is a part of. I'm going to talk about some challenges we have in studying phytosphere microbiomics, and then finally I'm going to add some extra complexity to the system as Mads was nice to present to me. So the Matrix is a large project, and 4 different universities are involved in this. We have a University of Copenhagen, which is the major partner, and then we have North Carolina State University, Technical University of Denmark, and then we have the NEO in the Netherlands involved. And if you take a look at the expertises of the different people, they have very different expertises, ranging from phenotyping to chemistry to strict bioinformatics, and also to into deep learning, and of course a lot of microbiologists are also involved. I'll get back to why the range of expertises are very important. We also have some imaging personnel on board. So as I said, Matrix is part of a very large grant granted by the Novo Nordisk Foundation, 203 million Danish kroner. And it basically consists of 3 large projects, where Matrix is the top one. And we have the Interact project run by Mette Nicolajsen, and the InRoot project run from Aarhus University. And so the matrix budget is 62.5 million Danish kroner, which is quite a substantial part of this. The two lower projects on the slide are actually studying below ground interactions, whereas matrix is focusing on the above ground biology. Okay, so one thing they have in common, all of them, is that they are focusing on wheat, which is an extremely important crop, of course. And just a fun fact that I found out that in Denmark, we have a 10,000 or 1,000 kilograms of wheat produced per person per year. And of course, as Mads was saying, there's a big interest in closing the yield gap in agriculture by adding microbial products. So this is something that's already taking place and something that some of the Danish companies are very interested in. So it's a common knowledge today that microbiomes are extremely important for the resilience and of course just life in general for eukaryotic organisms. And a lot of studies have been made that demonstrate where microbiomes are present in the body and also in plants. And most of these have focused on the so-called engine room, which is the root system, and also the human gut, as Tom was talking about, which is where most of the interactions are believed to actually take place. But I want to argue that it's very difficult. These systems are extremely complex. Tom was showing us one way around this, that the host genome is a big partner in this. But it's extremely complex, and I just want to show a few things about this. So if you take organism A, which produces a set of metabolites, it will influence the plant in a specific way to produce another set of metabolites that then again goes back and influences the microbe in which metabolites are produced. And if you have a second metabolite, you will get a second set of or a second bacteria. You will have a second set of metabolites, and that will influence the plant in another way. And if you have two bacteria together, they will influence each other to produce a third set of metabolites that then again will make a different response. And this is just under one condition. So let's say 4 degrees. So if you change the temperature, it's a whole new set of metabolites. And of course, Tom may not have this problem in studying gut microbiomes because it's a fairly standard temperature. But going into the field, we have all kinds of temperature ranges and UV, different UV radiations, moisture, etc. that varies along the way. So in order to get some reduction in simplicity, we are focusing not on that's not the only reason why we're focusing on the phylosphere, but also it's not much is known actually about this. But we reduced the phylosphere to just one leaf, and there's several reasons for us to do that. Well, first of all, the flag leaf of wheat is supposedly responsible for about 40% of the wheat yield in the end. So it's a very important leaf, and second of all, it has a fairly limited microbial and metabolome space. So for us to get a full systems description of this, we selected something with a limited space like this. The third reason is that we can actually, from the flag microbiome, from the flag leaf microbiome, we can culture basically, or at least most of the organisms in here. So that is also known from, and this is going to be very important, as I'll show later. And then again, it sticks out, and we don't need to go inside the plant or dig up the roots to actually study this using cameras. Okay. So actually, so when you think about this, we, in a nutshell, we have a flag leaf scenario. We have a bunch of microbes that we don't really know that well yet. They will influence each other, and they will influence the plant to produce a set of metabolites that you see on top, and this will make the plant or produce a phenotype that either gives a big yield or a smaller yield or something else. And this brings me to the main objective of the Matrix project is to make a predictive unsupervised deep learning model describing the connections between chemical, physical, biological parameters in a full system. That's why we aim for a reduced complexity. Okay, so in a nutshell, the plan for the Matrix is to focus on one leaf. And as I was joking when we had the kickoff meeting and people were introducing complexity and hindrances for doing this and that and this, and then I said, guys, this is one leaf. We have 62 million. So if we can't do this, we're out of business. So so I think when I say one leaf, it's of course done about 5,000 times with all the different parameters. So I'll just get back to why this is important. So we start in the matrix by having a mapping phase. This is where we're at now. So we have 100 and some plots in Denmark and 100 and some plots in North Carolina that we are sampling from. And so we're getting many samples from these fields, and we're going to do this for a couple of years. From each plot, we're going to do microbiomics. I'll get back to how we do that later. We're also going to measure the metabolome, and these are volatile compounds, non-volatiles, polar, apolar, and we have some smart chemistry people to actually take a look at this. And then we're going to do imaging of all the samples also, and these are both hyperspectral images, LICOR, and nutrient measurements, etc., for each of these samples. And furthermore, we're going to take existing mechanistic models that have been made for this and incorporate this into this unsupervised deep learning model. Now, I don't know a lot about deep learning, but I've been told that this type of model thrives on chaos. So that's what we're going to give it. We're going to give it thousands of samples with very many different variables, and we're going to see if it can actually predict something, how these variables are connected. So in the second half of the project, we're going to do a validation period or experiments in greenhouses where we're going to demonstrate if the model is right, and if it's not, we're going to use that data to put into the model again and evolve it in this way. Okay, so the microbe part of it is of course a lot of DNA sequencing, as you're looking at on top on thousands of samples. This has some problems. I'll get into that later. Furthermore, we're going to build a biobank of both fungal isolates, bacterial isolates, and also viral isolates, bacteriophages from this environment. And we're going to go from having small pieces of DNA, building gene catalogs, and actually building whole genomes from this environment to actually study the whole process. So the dream is to know the full genome or microbiome genome of the flag leaf, and also the chemical space. So when doing shotgun metagenomics on the phylosphere, it's quite tricky. If you want everything, all the microbes on a flag leaf, even just one leaf, you're going to have to crush it. And if you do that, you get 99% wheat DNA, which is not easy to get rid of, actually. And if you want a 10 gigabase shotgun library from this, you're going to have to sequence a terabase at least from each sample, and we can't do that for thousands of samples. So we have come up with several washing and filtering techniques to reduce this, but we're also compromising a little bit on the ones that are inside the leaf. So that's important. And alternatives to this is Amplicon sequencing that we can actually do on this. So the idea that if we can't get enough sequence out of this system, we want to go really deep and know all the genes and their connections by making a huge only flag leaf database of microbes that we can, even with small pieces of DNA, we can actually by statistics link this to a full phenotype in the genomes that we're investigating. Another reason why it's good to have microbial isolates, of course, you get the full genomes after sequencing, but you can also measure the metabolites that each microbe can produce under different conditions, and you can actually add them back into the to the field as we're going to do in the validation experiments. When we have the different components, the different metabolites, and the different microbes, we can actually manipulate the system to show if we're right. And of course, you can also look at genetic evolution by comparative genomics, as we did in this paper, where we looked at the evolution of different photosynthetic genes on bacteria that are actually sitting on the flag leaf. It's a very good site to sit in if you want to do photosynthesis, of course. So we already started to optimize the methods, and we did this by making a pilot or sampling last year, where we're actually sampling 51 sites on a field, and we're doing both hyperspectral imaging, all the metabolomics in the book, and also microbiomics on this, and we're also cultivating. This is just work in progress, but one way this could look is actually, so these are some of the principal metabolite components that we have mapped and can actually put on the field. And we're overlaying now hyperspectral images on this, and then microbiome data to see if we can find some correlations in that. We have also started to learn the system, to know the system a little bit better, because how many microbes are actually on one leaf, and we have made some numbers on this that are ranging up to between 10 to the 8 and 10 to the 9 bacteria, and we have optimized our sequencing methods, our Amplicon methods, to see how most efficiently we can describe the bacteria on there, and we found the major phyla. I'll get back to that later. We have also done a lot of cultivations, and we can see that we can indeed, both by targeted, go for the most dominant ones, and we can also non-targeted get a lot of microbes out of single leaves using these techniques. And just look at the colors here from this. These are from the NEO partners. And this probably is also related a little bit to the UV light in this. Okay, so one of the things that I'm getting there, Mads. So one of the things that we are looking at in our research group is the relationship of bacteriophages to microbes. And just in a conceptual nutshell, as we have discussed, eukaryotes are regulated by microbes and dependent on these. Sometimes, if you had a random system, some microbe would take over because, they're very competitive and one is better than the other at a specific point. But bacteriophages can actually, these are the regulating systems that keep things in check and keep the microbiomes balanced. Okay, how is this in the wheat phylosphere? Because this is a UV shower, and vira are notoriously known not to like UV light. So this has even been tested by several researchers that want to apply phages to the phylosphere to cure bacterial diseases. But these are very sensitive to UV light normally. So what we did was we took 500 flag leaves and some other leaves also, that's not important now, and we split it into a viral part and a bacterial part, and then we analyzed these. We did extraction, and you have to do some enzymatic and some filtering steps to actually get to the virome and separate them from bacteria. And this was a very exciting moment in the research, because did we get anything out of this? And we actually got a lot of DNA out of these samples, and if true, and that's the calculation below, we have a 0.154 nanograms of DNA per leaf, and if this is all viral DNA, we have a lot of bacteriophages. We have 10 to the 7 bacteriophages. And the analysis actually showed that 82% of this, at least, was viral DNA. So we were very content about this. It's very hard to know if your viromes are completely assembled, so ignore the rest of the data. And the next figure is going to be quite complicated. So the only take-home message from this is the phages that we found in the wheat virome, the vira from there, related to all the vira in the database. So these are protein similarities. And the take-home message from this is the wheat virome in yellow it has of course relations to other vira, but there's a lot of vira here that we don't know, and it's not surprising because it's actually the first phylosphere virome that has been studied. So we're very content about this. We can also actually relate the bacterial part of the phylosphere microbiome to the vira involved, or at least to a few of the vira involved. And what you can see on this picture is that most of the bacteria families are covered by vira that actually target members of this family. So the final slide here is just a picture of the most abundant one. It's very hard for me, but I made an arrow there. Approximately 20% of all the vira here is this phage, and this phage is extremely interesting. It's hosted normally in a bacterium that's inside aphids, and it protects the aphids from getting killed by wasps, and it does so by producing an insect toxin. So there's a whole story here that we haven't taken a look at, but bear with me, it's going to come out at some point. Okay, so just the take-home message from the last phage part here is that the phages actually also shape the phylosphere microbiomes, which was a relief, maybe not a surprise, but a relief for me. And one flag leaf has about 10 to the 8, 9 bacteria, and there are at least 10 to the 7 bacteriophages that hover over these and regulate them, and therefore we certainly believe that bacteriophages play a major role in shaping the plant microbiomes, as well as, of course, the human microbiome as it's known. So with that, I will say thank you to the organizers and to the funding, and also to the Matrix team and my research group. Thanks.
Great. Thank you so much, Lars. Very exciting talk and exciting topics, and yet another very complex project you are handling. But thank you so much. We have a couple of questions from the audience online, but keep asking questions. That'll be great, so we can get the dialogue and the Q&A running. Otherwise, you'll just have to listen to my stupid questions. But let's start with Louisa Svensson, who asked, this is for Tom. If you find the optimal genotype that responds best to a given feed, do you foresee that we in the future just will cultivate clones of the same genotype?
Yeah, that's a really good question, and I think there is a huge danger in that idea because, as we know, things evolve fast and change fast. But I think it's also going to be environmental dependent. So consider our fish, for example. We're studying fish in the coast off Bergen, but there are salmon rearing pens the whole way up the Norwegian coastline, and almost certainly in the different environments, different things will happen. And this goes back to Lars's point about how complex it is. So So whether it's practical is one question, but at least by doing this, we can start to explain things that we see. For example, it may be that you have a certain probiotic that you think works amazingly on chicken, and then you go and try them somewhere else and they don't work, and you've got an upset farmer saying, like, why is this probiotic not working, right? I mean, it will allow us to get this bit of sensitivity. And if you look at the human example, maybe, for example, this amazing work now on fecal microbe transplants, right? I mean, they have some awesome effects, but not on everybody. Why not everybody? Could it be because the genetic variation is playing a role there? So going back to the question, I don't think we can have one clone everywhere because the environment is complex, but that doesn't mean we're not in a system where we can start tailoring things, especially when we're in very controlled environments like greenhouses or growth houses and so on.
Okay, great. Do you have a comment, Lars, in the same area?
No.
Okay.
No.
Cool. Well, then I have another question for you. Yeah. It's from Michael Gilbert. Lars, would you consider soil, water, and air microbiome having impact and chance of traceability in the leaf genome?
That's a very good question. There has been a few studies that actually show that endophytes can be transported from the root system and the soil and into the phylosphere. So certainly that I see that happening. But it's probably as Tom was saying, it's probably what happens in the phylosphere is probably a permissible range of microbes that are actually very successful there. So I do see that you can separate the two by the core microbes that are there, but transiently there will be some transport in the system.
Okay, great. So back to some of related to both of your talks. So how important is it? You're studying cultivated plants and cultivated fish, so it's one genotype. But how important is it to also study the wild, to learn from that and translate that into our more cultivated systems?
Yeah, do you want to go first, or shall I?
Sure, I can, but I'm almost handing it over to you, Tom. So Tom and I had a dinner with the Thomas Jefferson at some point.
Richard Jefferson.
Richard Jefferson.
Not the former president of the US.
Not the former president, Richard Jefferson at some point. And he was giving a presentation about the hologenomics, and he's very convinced that of course in the wild, we built an artificial system in the agricultural system that is not the original host. But maybe Tom, you can elaborate a little bit more on that. So
yeah, I mean the problem with the wild is the environmental variation is huge, so it's really, really hard to see what's going on. And so from first principles, you have to start in a controlled system. So for example, we have actually done some plant experiments. We're not just fish people, but classically we will grow, for example, 6 or 10 lines of maize in a greenhouse with the same soil and everything, right? To look at the variation, right? And then from that you can go to the wild and see, well, does the pattern hold up? Do we see all that genetic genome variation noise just vanishing because the environment is so great? But to allude to what Lars was saying, I mean, it's a really good point that this guy Richard Jefferson raised, which is as we select on any line and we narrow its gene pool, if the genome is in some way shaping the microbiome, we are narrowing down the range of microbes that can work with it. Now, this is a problem. If, for example, you are surveying the wild and you find there is a wild teosinte related to maize and it's got some microbes that appear to be giving it a protective benefit against the pathogen, doesn't mean you can take that and throw it on domesticated maize B73, right? Because the genome's changed. So you have to start teasing these apart. But the good news is we can start to do that now, right? It's all about lab methods getting better and sequencing getting cheaper and computers bigger.
And if I could just add to that, a part of the chaos that we will also try to throw into this deep learning model is and I don't think I made that very clear. So we have several genotypes both in Denmark and in North Carolina, and are also planning to do wild ancestors of wheat that just to see any correlations in that.
Great, great, thank you. So we have an anonymous question here. It's related back to some lab work. How are you quantifying the plate experiments? What type of data are you getting from it? plate data, plate experiments.
How we are quantifying so we are simply counting the CFU, the colony forming units so far. So we are not planning. So just maybe that this was not extremely clear. So we are not doing cultivation for each of the thousands of samples, because I mean that would require an additional 62 million just to have somebody do all the cultivation part of it. But we are planning to cultivate compiled samples to basically make sure that we cultivate all the microbes that are that cover the variation on the flag leaves overall. Yeah.
Then a phage-related question, Lars and Tom, of course. Could you speculate the source of philosphere phage? Do some of them actually come from insects?
It's a very good question. I'm sure they do. We also know that a lot of them come from inside the bacteria, which are induced prophages. But there's so many things we don't know in this environment. I mean, normally a way to induce a prophage in a cultivated bacteria in a lab is to put UV light on it. So that's going to make an explosion of a prophage induction into the lytic phase normally. But if that is more significant in this, or if the bacteria and the phages have adapted to the high UV, we don't know. So yeah.
Great. So Tom, here's a question. Maybe you talked a little bit to it before, but this is from Michael Goray again. There are many plans for monoculture land-based fish production. How would you see this environment change microbiome?
It will absolutely change the microbiome. There's no doubt about that. But actually, it's awesome for our hypothesis, because you're getting an even more constrained environment. When you're growing fish at sea in sea pens, of course, there is current going through it and stuff, right? And big storms will change things. But once you're growing them on the land-based situation, it is actually the perfect situation for a situation where you may want to actually select on genotypes for a microbiome. So. And I think that's the point. The resistance against the idea of the genome being important, it's largely based on observations in humans and wild things, which is fair enough. But that's not where most of our food comes from today, right? So the more constrained things get, and if you start thinking about hydroponic or aeroponic crop production, for example, it's a similar situation. So this is where it's going to get more and more important to personalize it.
And if I can add to that,
sure.
I think also by reducing the complexity, which is a necessity to produce large amounts of fish or wheat and so forth, and putting things extremely closely together, you're also making an environment that's quite vulnerable to diseases, both bacterial and viral diseases that can spread in these. And simplifying the microbiome, and thereby maybe making it less robust.
So we have a comment here from Rupinder Kaur. For Tom, wonderful talk. What is the future of personalized probiotics or fecal matter transplant in the wildlife conservation field? Would it be helpful?
Yeah, well, that's the future depends on if anybody takes me seriously who's got any money, for a start. But beyond that, it's a really great question. I mean, it comes down to, do we actually think in the long run things like microbiomes are a solution to some of the big problems we're facing, right? So obviously in human health, yes, microbiome therapy is important, because when you're treating certain diseases, nothing else is working. In the wildlife conservation context, I think more of a problem is people chopping down trees or polluting the environment, and there the microbiome is probably not going to do very much, right? So it could be something useful, but probably it's going to be irrelevant versus changing policy and these kind of things.
Thanks. Then we have a question relating to deep learning, Lars, and great talk, she says, Celine Krone. What were the benefits of using deep learning in these studies, or what will the benefit be?
I think the benefit of I'm not a deep learning expert, but as I understand, the benefit of doing this is to utilize the chaos that these types of models actually thrive on. So I think the most simple way to put that is that if you have a system that's seemingly chaos, and you can of course you can put some hypotheses in and test whether this or that or this component is important and will influence these other components in this way. But if you have a system that's this complex, I think I'm convinced, or somebody convinced me in the project that this is actually a way to go forward. Yeah.
Great.
I think it's the only way. When you look at the size of the data sets, there's not much choice.
No. Yeah. Even with a reduced complexity as the flag leaf. But,
yeah. Another question for you, Tom, from Tine Rask-Licht. Tom, thanks for a great talk. Which traits encoded by the host genome do you think are the most important for determining the microbiome phenotype?
Yeah, that is an actually awesome question, but I think actually the answer is there could be so many. The beauty of the fact that the environment does have an effect on the microbiome is that there are so many ways you can change the environment in an animal or a plant's body genetically. For example, consider humans, right? If we change our pH of our saliva, right, or the enzyme concentration, or even probably the length of our tongue, or the pH of the gut, or the length of the microvilli, right, or even the temperature of the person, or the dietary preference, all of these will affect the community, right? And that is why, to me, it is completely unsurprising the genome must be relevant, right? So a very, very simple example, people in this part of the world can drink milk. People in lots of the other part of the world can't drink milk. Just that alone, that one genetic effect on the milk will change the microbiome, right? So I think actually the beauty is there are so many ways it could happen, and that's what we'll find out when we start looking in detail.
Great. Thanks. So for generating all these high-quality data sets, what's the greatest research challenge to actually get access to all these data?
Yeah, I mean, do you want me to go? You?
Oh, sure, go ahead.
Okay, so actually the biggest challenge is consistency. It's very, very clear from microbiome studies, but also transcriptome studies, metatranscriptome studies, and so on, that if you collect samples and store samples in different ways and process them in different ways, you introduce biases, right? And that actually makes it a nightmare, because you might have done a study 5 years ago, and then you might want to replicate it, but the methods aren't even the same because you can't buy them anymore. So actually, what we're encouraging is think to the future now. When we're doing these massive salmon or chicken trials, which are costing huge amounts for people to set up, oversample, spend the extra money on extra people there to sample and store for the future, as opposed to then coming back and start again in the future, because those little lab effects have huge downstream effects and can make life very difficult.
Yeah, and if I can, I think that's excellent learnings from museums and all of that, and I also think that it's very crucial to be aware of the biases that are in your system, and make all the controls you can do to compensate for that, but they will all have biases, and therefore it's also important for us to, within the three larger projects in the collaborative crop resiliency program, to align our analysis methods. The cultivation is very easy to align how to do that, but how we do DNA extractions, how we do even the sequencing is giving a huge bias in one way or the other. If you do Amplicon sequencing, it's primer choice and all of that's also giving completely. But even what sequencing platform you're using, if you're using Illumina or Nanopore or that will change the microbiome completely. So it's many things to consider.
Great. I think we have to wrap up. Thank you so much,
Thank you,
Tom, Lars. It was great spending the afternoon, and thank you for coming here and presenting your great research. Thank you so much.
A pleasure.
Thanks.
And thank you for all the good questions and great answers.